Beyond the type, how genetic switches unlocked a 50-year blood mystery for decades, a fundamental question puzzled hematologists. Why do two people with the exact same blood type possess vastly different levels of protective molecules on their red blood cells? Researchers at Lund University in Sweden have finally provided the answer. By looking beyond the genes themselves and focusing on how those genes are switched on and off, scientists have uncovered a hidden layer of genetic regulation that could revolutionize transfusion safety and our understanding of disease resistance. The hidden layer of blood chemistry. When we talk about blood types, we usually think of the standard A, B, AB, or O categories. However, blood compatibility is much more nuanced. It depends on antigen-specific molecules sitting on the surface of red blood cells that act as biological ID cards. The problem is that the quantity of these antigens varies wildly between individuals. If you only have a couple of hundred blood group molecules per cell instead of 1,000 or even 1,000,000, there is a risk they might be missed in a compatibility test, which can affect the safety of a transfusion, warns Martin L. Olsen, professor of transfusion medicine at Lund University. Standard DNA tests often fail to catch these discrepancies because they look at the blueprint, the gene, rather than the volume knob, the regulation. The discovery of molecular switches to solve this. The research team shifted their focus from the genes to transcription factors. These are specialized proteins that act as molecular switches. They bind to specific regions of DNA to dictate how much of a particular protein a cell produces. Using a sophisticated new computational pipeline, the team mapped nearly 200 of these binding sites across 33 different blood group genes. This allowed them to identify why certain genes, despite being present, were barely functioning. Solving the Helgesen mystery, the breakthrough provided an answer to a legendary medical anomaly known as the Helgesen blood group. Discovered in the 1970s by medical technologist Margaret Helgesen, this rare variant is characterized by extremely low levels of a protein called complement receptor 1, CR1. For 50 years, scientists couldn't find the genetic cause, even when testing the DNA itself. The Lund University team discovered that the issue wasn't a broken gene, but a broken switch. A tiny mutation in the DNA prevented the necessary transcription factor from attaching to the gene. As a result, the gene simply idles, producing far fewer molecules than normal, an evolutionary trade-off. Protection versus risk. This discovery also highlights a fascinating intersection between genetics and evolution. The study found that this specific low CR1 variant is more common in Thai populations than in Swedish populations. There is a biological reason for this. Lower CR1 levels appear to provide protection against malaria. By making it harder for the parasite to invade red blood cells, the mutation offers a survival advantage in regions where malaria is endemic. This illustrates a classic evolutionary trade-off, where a trait that complicates modern medical procedures may have been a vital shield against ancient diseases. The future of transfusion medicine, the implications of this research, extend far beyond a single rare blood group. The team's data-driven approach is already yielding new insights. Improved diagnostics. Researchers are working to update DNA-based testing chips to include these newly discovered regulatory variants, making blood matching significantly safer. Expanding the map. Follow-up studies have already identified similar regulatory issues in the crucial RHD blood group, explaining why some patients show extremely low levels of the protein despite having normal genes. A new research model. By combining computational tools with epigenetic data, scientists can now predict how different blood groups might affect a person's susceptibility to various diseases. Conclusion. By shifting the focus from gene sequences to genetic regulation, scientists have bridged a 50-year gap in blood science, paving the way for safer transfusions and a deeper understanding of how our biology protects us from infection.